Dear students, this is our 27th lecture on bioorganic chemistry, and today we will discuss about artificial enzymes. As you know, the natural enzymes, they are very effective for catalysis of different types of natural processes, and because of that the scientists, they have imitated these natural catalysts, that is enzymes. So the mimics of these natural catalysts, they are called as artificial enzymes. And this field was first studied by Ronald Breslow, and two terms they have been used for such type of artificial enzymes: biomimetic chemistry or artificial enzyme. So biomimetic chemistry is defined as a method of scientific inquiry that involves mimicking the laws of nature to create new synthetic compounds. Therefore, a biomimetic compound is a compound that mimics a biological material in its structure or function. So, if we synthesize a compound which have properties or structure or function similar to the natural enzymes, then that compound will be called as biomimetic compound. And that process through which that biomimetic compound has been synthesized, that will be called as biomimetic process. So a laboratory procedure designed to imitate a natural chemical process is called as biomimetic process. So there are few examples. For example, cyclodextrin dimers, they have been studied as antibody mimics. So if two cyclodextrin rings, they are linked to form dimers like this, then these dimers, they can have strong binding properties, and they can bind natural materials such as steroids. What happens is that in case of one cyclodextrin molecule, the size of cavity is 7.8 angstrom units. Whereas for a steroid like cholesterol, the length of the molecule, say here, is 15 angstrom units in its length. Now, if this is 7.8, that means if we join two cyclodextrin molecules, we will have a cavity which can fit these types of steroids into it. So therefore, that is why these cyclodextrin dimers, they are good for selective cholesterol binding, and we can use a very small type of linker to bind these two cyclodextrin units. And sulfur is single sulfur atom is used for this purpose. So when two molecules of cyclodextrin, they react with one molecule single sulfur atom, then these types of complexes, they are formed. So two molecules of beta cyclodextrin linked by single sulfur atom, they will have a cavity which will selectively bind cholesterol. So we can synthesize these type of complex as antibody mimics. Then another example is acylation of cyclodextrin. Uh, we know that beta cyclodextrin, they have these hydroxyl groups, and this hydroxyl group present at carbon 2 of the glucose molecule, This will act as a substrate for esterification, acylation with meta-nitro uh, benzyl acetate, meta-nitro phenyl acetate. This will react with beta-cyclodextrin to give us these types of cyclodextrin esters. And it has been shown that approximately a rate of deacylation of this substrate increases 100 folds when it reacts with the beta cyclodextrin. If we compare this with the hydrolysis under same conditions, here only because this substrate, this could bind well into cyclodextrin cavity, and a tetrahedral intermediate formed during acylation fits into the cavity of the beta cyclodextrin very easily, and because of that the reaction is 100 times faster than the normal acylation reaction. Similarly, in case of ferrocene esters, it was found that they will bound into the cyclodextrin cavity, and acylation will take place at about 60 lakh folds more in relative to the hydrolysis of these esters in the absence of cyclodextrins. So here also the tetrahedral intermediate formed in the transition state fits into the cavity of the beta cyclodextrin so easily that the reaction becomes about 60 lakh times faster than the normal hydrolysis reaction. Then many enzymes, as we know, they use coenzymes which act as catalysts in biomimetic chemistry. For example, we know that thiamine pyrophosphate coenzyme is used for decarboxylation of alpha keto acids. And when gamma cyclodextrins they are attached to the thiazolium ring. Thiazolium ring is present in thiamine pyrophosphate also. So we can produce mimics of this coenzyme by attaching this thiazolium rings to the gamma cyclodextrins, and they will act as mimics of thiamine pyrophosphate. So the most common example of using these as mimics for benzoin condensation. So two benzaldehyde molecules, they are catalyzed by this mimic of thiamine pyrophosphate to give us benzoin as the product. Then another mimic is formed from beta cyclodextrin with this pyridoxamine moiety, and we know that pyridoxal phosphate and pyridoxamine phosphate, they are used for transamination of amino acids. So this mimic of this pyridoxal phosphate and pyridoxamine phosphate is used in combination with beta cyclodextrin for this type of transamination reaction. And when this pyridoxamine moiety in combination with beta cyclodextrin is used as a mimic, that reaction is approximately 100 folds more than in the normal circumstances for the transamination of phenyl pyruvic acid relative to pyruvic acid. And if we use the 4-tertiary butyl phenyl group, this is 4-tertiary butyl phenyl group, then this reaction is 15,000 times faster for the transamination reaction. So this can be used effectively as a mimic of this coenzyme. Then there are some other example. Enzyme ribonuclease A, we know, is used for hydrolysis of RNA, and the active sites on this enzyme, they are histidine 12 and histidine 119. So if we can prepare a mimic having two histidine moieties present on that mimic, then that can also be used for hydrolysis of different type of RNA. So therefore, several cyclodextrin bis-imidazole mimics, they have been prepared and studied for their ability to catalyze the hydrolysis of a cyclic phosphate esters, which is the process is the same as carried out by enzyme ribonuclease A. So we can see over here there are different combinations which can be used for these types of enzyme mimics. So one is when two imidazoles, they are placed on adjacent primary carbons of the glucose, that is A and B, adjacent uh, primary carbons of the glucose, beta cyclodextrin. One complex is formed like this. Another combination can be when two imidazoles, they are placed on primary carbons of glucose A and C. So one glucose atom away from this, then another atom away from this glucose A and D. So different combinations of imidazole moiety is used for different types of mimicking reaction. So when this cyclophosphate ester is hydrolyzed by these types of moieties, this can mimic the same enzymatic process which has been done by ribonuclease A enzyme. So this beta cyclodextrin bis-imidazole mimics the phosphate ester hydrolysis very effectively. Then another cyclodextrin dimer which is based on 2,2-bipyridyl group. This is the 2,2-bipyridyl group which is acting as a linker to this cyclodextrin molecule. This will permit to incorporate a catalytic metal ion. Because of the presence of these two nitrogens, these will act as a ligand. So copper 2 plus ions can bind to these nitrogens, and this complex will be used for hydrolysis of an ester. So when this mimic was formed, that it was seen that the reaction is 2,20,000 folds faster when this compound 4-nitrophenyl 3-adamantane 1-alpro-2 ionoate is used as a substrate for its hydrolysis. So hydrolysis will give carboxylic acid and para-nitrophenol as the product. But this reaction is this much times faster than the normal reaction, and it was found that copper 2 plus ions, they act as a better catalyst than zinc 2 plus or nickel 2 plus ions. Thank you very much.